Um, Mona, it's a graduate student in our lab, and she started about 5-6 years ago, and has demonstrated an amazing capacity to teach others, to educate, to a tremendous creative spirit in her research, as you will soon see, and very sophisticated methods and analysis that she has developed and carried on, important assays to study pain and across the human population, across all ages. Mona will be continuing with us under a prestigious fellowship from the Nancy Lurie Marks Family Foundation, so that she continues this unique line of research, because it doesn't exist anywhere else. It's very unique, and so she was funded to continue on with it. And in addition to that, she has, in her very altruistic spirit, she has developed courses to educate the population about these internal states of people who have distress and suffer from pain and from neurodevelopmental disabilities. And so starting this year, already we're in 2024, will be deploying a certificate program to that end, and she will be spearheading that in parallel to her scientific endeavors in the lab as a postdoctoral fellow. So let's listen to Mona's dissertation and enjoy. Go ahead, Mona.
All right. Hi, everyone. Welcome. Okay, so today I will be presenting to you my thesis work on developing biometrics that are interpretable in terms of understanding individuals' internal states of anxiety and distress and pain, with a particular focus on the autistic population, but can be applied to the general population as well. So to start off with some background motivation behind this work, a lot of studies that look at human behavior and cognition have primarily focused on the activity of the brain, the top-down efferent signals from the central nervous system and how that influences the body. However, we know that there's continuous communication between the brain and the body, where the peripheral nervous system sends continuous afferent or bottom-up signaling, providing feedback to the brain to guide future action and behavior. And the peripheral nervous system has two main branches: the somatic system, which controls our muscle movements, our sensation of the external world, and the autonomic system, which guides our body's overall homeostasis, such as like our body temperature, digestion, cardiac activity, respiration. So each of these play an important role in our kind of bodily awareness, our ability to coordinate our movements or express different emotions, our overall kind of internal states. So when looking at human behavior and cognition, it's important to assess both the top-down and bottom-up signals that influence our behaviors. So in this work, we pay particular attention to cardiac rhythms as a proxy for understanding the autonomic system. So as you can see in this illustration here, there are abundant connections between the brain and the heart, which guides our overall parasympathetic and sympathetic states of our nervous systems. sorry. So our sympathetic nervous system guides our body's overall kind of stress responses, states of fight or flight, whereas the parasympathetic system brings the body back to a state of relaxation. And the balance between these two systems can have cascading effects on aspects of our cognition, our ability to regulate our behaviors and our emotions. And you can imagine someone who is in a state of anxiety and distress, having trouble kind of guiding their cognitive resources, focusing their attention to kind of learn or make decisions, or even kind of engage with other people, or reciprocate kind of social interactions. And this becomes especially important when looking at the autistic and other vulnerable populations. So we know that individuals with autism may experience high sensitivity to certain sensory and pain stimuli, and their pain experiences often go unnoticed and undertreated. And the main issue here is that most of the pain assessments that exist rely on kind of verbally reporting how the individual feels, or using these visuals or adjectives to describe the pain that they're experiencing. And this can be very limiting in terms of, people who may express their pain much differently than the neurotypical individual. So we propose using kind of the underlying physiology of the person, their cardiac rhythms, their movement patterns, their facial expressivity, to better understand kind of these and characterize these internal states, to obtain kind of more objective assessment approaches. So in this work, we take an integrated approach where we look at a variety of internal processes, such as our cardiac rhythms, using EKG or PPG sensors, and other aspects that are kind of more externally observable, such as our movements, our facial expressions, our eye gaze. And we use kind of motion sensors and video-based kind of motion tracking to kind of understand these subtle changes within these externally observable processes, because there can be changes within them that can go undetected to even the trained eye. So we incorporate these biorhythmic kind of signals with a variety of motor cognitive tasks, as well as scores on questionnaires and assessments, to gain a more holistic kind of understanding of internal states of pain and distress. And then we take a personalized analytical approach inspired by precision medicine, where we first look at the biosignatures of the individual, and track, common patterns or trends that allow for clustering or grouping within our sample. And this ultimately allows us to kind of stratify the population based on the similarities that are observed. All right, so here is kind of the overview of the thesis. We have 3 main studies. In study 1, we look at the cardiac, motor, and facial responses to physically induced pain in neurotypical individuals. We then extend that to the autistic population, and ultimately assess kind of the real world applicability of these approaches. And in this work, we take a common statistical kind of pipeline to assess all of the biorhythms that we're capturing, so across the face, the heart, and the body. So for example, if we're looking at a motor signal such as acceleration in 3D space, we take the Euclidean norm of this and convert it to micro movement spikes that control for anatomical differences across participants. And this can be applied to the cardiac signal as well as the facial landmarks, as well as eye gaze information. And based on empirical estimation of these biorhythmic signals and past studies within the lab, we, using maximum likelihood estimation, we find that these biorhythmic signals fit best under the gamma probability, the gamma family of probability distributions, where we obtain useful kind of parameters such as the gamma scale and the gamma shape, as well as other gamma moments to characterize these biorhythmic signals. So here we take a closer look at these gamma parameters as they'll come up frequently when looking at the results. We have two main parameters, the gamma scale, which represents the gamma variance over the gamma mean and the overall kind of dispersion within a probability density function. So you can see here in this illustration how higher gamma shape values lead to more high gamma scale values lead to distributions that are wider with more dispersion, whereas lower scale values lead to more peaked distributions. And this is how we obtain it kind of computationally. And then the gamma shape parameter, which represents the overall shape of the distribution, whether it's Gaussian or more symmetrical. And then lower gamma shape values are associated with more skewed distributions with heavy tails, with gamma shape being one, demonstrating an exponential distribution. And here is kind of an illustration of the theoretical kind of probability density functions based on the shape and scale values, and based on several studies across 2 decades in the lab, we find kind of clinically interpretable kind of meaning to these biometrics, where we find that the gamma scale value represents kind of the overall noise to signal ratio within the biorhythmic signal. So parameters that fall in the left kind of upper region here demonstrate signals with high noise and uncertainty, whereas the parameters with higher gamma shape and lower gamma scale values represent signals with increased predictability and decreased noise. And we find that these two metrics are inversely proportional, and this has been used to assess developmental stages and characterize a variety of neurological disorders and illnesses. All right, so we'll start off with looking at study 1, where we assess the biorhythmic signals and how they change in response to pressure-induced pain in the neurotypical population. So a bit of background here. Pain alone is an intrinsically undesirable experience. It can lead to tissue damage, but is also associated with sensory, cognitive, affective, and social components. It's a leading contributor to disability and the opioid crisis. And the current issue with pain is that most of the assessments out there rely on kind of discretized subjective reports. So the goal of this study is to kind of obtain a more objective kind of assessment of pain based on the kind of internal physiology of the person, and kind of integrate that with some of the self-report metrics that exist out there. So in study 1, we had about 45 neurotypical individuals, 27 of them female, and we utilized the standard blood pressure cuff on the arm to induce pressure pain. And then we have these BioStamp MC10 sensors that capture the motor and cardiac activity, as well as a webcam to capture videos of the face. And then we also asked participants to rate their pain sensitivity levels and the pain that they're currently experiencing throughout the study. So in the study, we have a variety of tasks. We have a baseline resting task where they sit in a relaxed position and avoid excess movement. And then we also have a variety of motor cognitive tasks. So the first one here is the drawing task from the Montreal Cognitive Assessment, where they connect a sequence of letters and numbers. And this looks at kind of memory and executive functioning skills. We have a pointing task that's commonly used in the lab to assess aspects of motor control. The participant kind of raises their arm to reach to a target, which is kind of an intentional movement. And then they bring their arm back to rest, which is unintentional. And this has been very useful in the past to characterize a variety of neurological kind of illnesses. And lastly, we have a peg task where these metal kind of pegs are rotated and inserted into these grooved slots. And this looks at aspects of fine motor skills. All right, so we have a variety of conditions where they perform these tasks. So in the first condition, it's kind of the baseline control where the participant does the task using their dominant arm. No pain or anything is induced. We have a second control condition to control for any learning effects during the peg and the drawing task only. And then we have the pain condition where we utilize the blood pressure cuff. We wrap it around the arm and inflate it. And this was a procedure introduced to us through our collaborator, David Yernitsky. So we inflate the cuff. We have them perform the activity. And then right before deflating the cuff, we ask them to rate the pressure pain sensation. And then lastly, we look at any after effects of the pain during the post-pain condition. All right, so first we'll look at some results from the movement signal. So for the drawing task, we saw the most distinct patterns when looking at the movement of the upper arm or the bicep sensor. So this represents the gamma noise to signal ratio of the movement of the upper arm, the acceleration micro movement spikes. And what we see is a general increase in the kind of noise during the pain and post-pain condition in contrast to the control, which is the blue here. And this difference is significant, mostly for the pain condition. And as expected, we see a decrease in the gamma shape parameter that's consistent across the pain and the post-pain condition, which suggests that the pressure pain may be interfering with the motor signal. For the PEC test, we observe a similar trend and with the most distinct changes in the gamma variance during the pain condition, where there's significantly more variability in the motor signal during the pain condition. And when we look at task performance in terms of how much time it takes them to complete the task, for the drawing, we see a high kind of learning component where the first control condition, they take the most time to complete the task, whereas for the remaining conditions, the timing stays about the same. However, when we compare this to the motor signal, we see that kind of the less time they take to complete the task, the lower the overall kind of noise within the motor signal, and vice versa for the gamma shape. For the PEC task, we see more distinct kind of increases in the time it takes to complete the task during the pain condition, which suggests that the kind of pain may be interfering more with performance on this task, or kind of the pressure-based haptic feedback that they receive as the arm is kind of in midair as they kind of take the pegs and insert them into the slots. Whereas in the drawing task, they receive kind of continuous haptic feedback from the pen on the paper. All right, so lastly, we look at the pointing task, and we see similar trends here as well, where the noise to signal ratio is elevated during the pain and post-pain conditions. And this is generally guided by an overall increase in the gamma variance, suggesting that, again, that the pain may be leading to increased noise and unpredictability in the motor signal. Right. So next we look at the cardiac biorhythms. In addition to the gamma approach, we utilize a variety of heart rate variability metrics. So for this, we look at the ECG signals and the R peaks, which represents kind of the beat, the heart rate, the heartbeat, and the timings in between these peaks, which represents the interbeat interval. And we look at this signal as an overall measure of kind of heart rate variability. We look at it in the frequency domain, where we take the power spectrum of the signal and look at kind of low frequency components, which based on the literature are associated with sympathetic nervous system activation, and higher frequency ranges that are associated with parasympathetic nervous system activity. And then we can also look at magnitude scalograms, assess how these LF and HF components change over time. And then lastly, the time domain metrics via the Poincaré plots, where we plot the R interval or the IBI interval against the subsequent interval. And we obtain the SD1 metric that's associated with parasympathetic activity, and the SD2, which represents kind of the width and the longer term kind of changes within the IBI signal and is associated with sympathetic nervous system activity or the stress response. So some results here, we see distinct changes in the gamma noise to signal ratio and the gamma shape across the control and the pain conditions for all of the tasks that are used in the study. And this is what it kind of looks like when we look at the gamma parameter plane. What we commonly observed was the pain condition led to elevated gamma scale or noise to signal ratio, which was associated with elevated magnitudes in the LF range of the magnitude scalegram, which signifies kind of sympathetic nervous system activation during the pain states. Whereas during the control conditions, we see higher gamma shape values and lower scale or gamma NSR. And this is associated with kind of a more balanced activities across the low frequency and high frequency ranges. Interestingly, when we look at a subject who reported experiencing acute and chronic pain post-experiment, we find a unique response in their cardiac activity with high magnitudes in the 100 to 150 millihertz range that was consistent across both the control and the pain condition. And looking at their IBI signal, we can actually see kind of the low frequency modulation. And when we plot their gamma scale and shape parameters, based on the 95% confidence intervals that are overlapping, we can tell that there's not much distinction across the control and pain conditions. So this suggests that the pain may be not interfering as much in this with the cardiac signal in this particular participant, or that the pain that they are currently experiencing due to their injury was kind of overriding the experimentally induced pressure pain. In fact, they did report that, you know, the pain that they were experiencing due to their injury kind of was a lot more intense than the experimentally induced pain. Right, so other metrics that we find kind of most distinctly change in response to the pain induction was the Poincaré SD2 metric, which is also associated with sympathetic nervous system activation. And here we can see how for most participants, there's an increase in the SD2 metric during the pain condition. And this was commonly associated with an increase in the gamma NSR. And so this close kind of these consistent patterns across these two metrics allowed us to kind of perform some parameter identification approaches where we found high correspondence between the SD2 and the gamma NSR metric that's consistent across the control and the pain conditions. And this is useful in terms of kind of parameter reduction when assessing these biometrics of the cardiac activity, and the fact that, you know, commonly used heart rate variability metrics kind of correspond with the metrics obtained from our gamma approach gives us kind of a better understanding of the clinical interpretation of these metrics. And lastly, we look at the reported pain ratings across tasks. We see that they're relatively consistent across the resting, drawing, and pointing and peg tasks. And this was usually associated with a high correspondence with the perceived pain sensitivity ratings. So these together tell us that the participant has kind of an accurate representation of what their pain sensitivity is like. And then in an attempt to correspond this with the continuous kind of cardiac biorhythms, we see a general kind of weak to moderate correlation between the discretized pain ratings and the LF power of the cardiac signal. So this suggests that the kind of self-report metrics can complement the biorhythmic signals that we obtain from the individual. All right, so lastly, we look at facial expressivity within this study. So for this, we utilize the video data and open-face machine learning-based facial landmark detection that tracks the facial landmarks, 68 different facial landmarks across the face, as well as eye gaze and head orientation information. And we analyzed the kind of movement patterns of these landmarks based on the trigeminal areas of the face. So the V1, V2, and the V3 regions. And then we also utilize transfer entropy approaches to assess kind of the directed flow of information between these three regions and understand how they can influence one another. And then lastly, we also look at facial action units, which are kind of the building blocks of our facial expressions based on the muscular anatomy of the face. So for example, action unit 1 is associated with raising the inner eyebrows, and open face, we can track the intensities, the presence and intensities of these units. So looking at the motor signatures of the face, we generally find high gamma scale values in the V1 region of the face, followed by the V3 and then the V2 region, which demonstrates kind of increased kind of variability in the V1 region of the face, which kind of corresponds with the transfer entropy ranges that are associated with how the V1 region influences V2 and V3. And interestingly, when we correlate the gamma NSR with the transfer entropy metrics, we see a high correspondence between them, between the gamma NSR of the V2 region and the transfer entropy between V1, V2, and V1 and V3, suggesting that overall, the V2 is kind of codependent on the movement of the V1 and the V3 regions, whereas the V1 and the V3 kind of operate rather independently. And then looking at the facial action units, we see kind of significant changes in terms of the intensity ranges of these particular action units. So AU9, which is associated with wrinkling the nose, AU10 and AU25, which is kind of movements of the upper lips. And then lastly, which is consistent across all of the tasks used in the study, AU15, which is lip corner depressor. And based on the literature, we see that these two kind of facial action units are associated with emotions of sadness and disgust. All right, so in study 2, we kind of aim to use these kind of biometrics that we discovered in study 1 and extend it to the autistic population. So in this work, we are motivated by kind of exploring autism in their responses to pain using more objective approaches. Most of the studies on pain and autism that exist out there rely on behavioral observation or qualitative data. And so more objective assessment approaches are needed to kind of detect states of pain and distress in these individuals. And this can lead to kind of earlier diagnosis and treatment. So in this study, we utilize thermal pain induction procedures. So study 2 was conducted by our collaborators at Haifa University, who had about 141 adults, 60 of them being autistic, and they utilized this E4 wristband to capture cardiac activity. And they had a resting kind of baseline task, as well as 2 pain tests, the habituation test, where this thermode was used to elicit heat pain stimuli on the forearm, and a conditioned pain modulation test, where the one hand was inserted in a hot water bath and the other arm kind of received the heat pain stimuli. They also utilized a variety of questionnaires and assessments looking at autism severity, IQ, and overall kind of disability, mental, physical, emotional health. All right, so in this study, we have just the cardiac activity that we analyzed, and also compare to kind of some of the discretized self-report assessments. So here is an example of the magnitude scalograms during kind of resting state, and how that changes during the habituation test, where the pain is induced. And you can see how the pain elicits kind of increased magnitudes in the low frequency ranges of these scalograms. And interestingly, when we look at these metrics across the each individuals, we see unique patterns based on their baseline levels of activity. So across the entire population, there's general increases in the LF power, or the sympathetic response, and decreases in the parasympathetic activity. But here, when we look at each individual, we see that during the pain condition, the habituation, and the post-pain or condition, the increases are in LF power are most clear in individuals who have a low baseline LF power, and the decreases in HF power are most distinct in individuals with a high baseline HF power or parasympathetic activity. Excuse me. And so this suggests that, the pain may be inhibiting parasympathetic responses or leading to elevated sympathetic activity. And interestingly, when we look at the magnitude scalograms for several autistic individuals, we see this unique band of activity that mimics what we observed in the chronic pain subject in study 1. When we look at their frequency domain metrics, the LF and the HF, across the entire sample, these differences are not as distinct, but you can see here comparing each individual, how there's generally increases in the LF power and decreases in the HF power during the pain condition. That's consistent across post-pain as well. But interestingly, when we use the gamma analytical approach and look at the noise to signal ratios, we see across both the TD and the typically developing and the autistic group, significant increases in the noise to signal ratio across all of the pain tests, and with some after effects of the thermal pain stimulation. And this is consistent with our findings in study 1, where we also see decreases in the gamma shape values across the pain during the pain conditions. That's consistent across the TD and the ASD groups. Which further suggests that, the pain may be interfering with the cardiac signal, leading to increases in the noise and uncertainty of the heart activity. And based on the strong correspondence between the Poincaré SD2 metric and the gamma NSR, we try to look at how we can differentiate between the ASD and the TD group using the fit between these two metrics. So here we obtain the delta values, which are kind of the prediction error or the distance between each point and the line of fit. And what we see is across conditions, there are significant increases in these delta values for the TD group compared to the ASD group. And these linear fittings between the two metrics further allow us to separate between males and females in each of these groups. So you can see here, we plot the delta values during the resting task against the values obtained from the pain or the habituation test. And we see that the males fall kind of in this region that is distinct from the females, and that their delta values change across both the resting and the habituation pains test. Whereas for the females, we see that they mostly change during the habituation test, with kind of separation between the female and the male autistic group, which is quite useful in kind of characterizing pain and distress in the female phenotype, where there's a 5-to-1 ratio in terms of between males and females in terms of diagnosis. So utilizing these metrics, we can gain a better understanding of kind of the physiology across males and females in an ASD as well as neurotypical individuals. And then we further try to assess differences between both groups using a variety of machine learning approaches, as well as incorporating the self-report metrics. So in the cardiac activity, we observe at baseline kind of elevated LF to HF ratios that is guided by a decrease in HF power in the ASD group, which suggests that at baseline, these individuals have inhibited parasympathetic activity. And then when we look at their scores on some of the assessments of disability and overall kind of health, we see that in the ASD group, there's generally increased scores on the disability assessment and lower scores on assessments of health and emotional, physical health and emotional well-being. And in an attempt to correspond this with the continuous kind of biorhythmic signals, we see that scores on general health from the SF-36 tend to negatively correspond with the gamma NSR values from the cardiac activity, which suggests that increased noise within the cardiac signals is associated with lower scores on general health. And using machine learning approaches and a variety of the assessment scores, as well as the cardiac biometrics, we are able to kind of separate between the TD and the ASD group and kind of predict which individuals belong to each group with 71.6% validation accuracy. So overall, this demonstrates how these physiological signals or the activity of the heart can be useful for characterizing the autistic phenotype and their physiological responses to pain. So lastly, we attempt to kind of apply these approaches outside of laboratory settings in neurotypical and autistic individuals. So this study was inspired by a lab in a box project where we decided we could no longer collect data inside the lab due to COVID. And so we would take our equipment and devices and send them to participants to collect data within their homes or outside of the lab. And this, overall kind of helped us increase the accessibility of our approaches. So those who couldn't easily make it to the lab due to physical or time constraints were able to perform our experiments. And this also gave us an idea of how well these approaches can be applied outside of laboratory settings. So in this study, we focus on the biorhythmic signals that we discussed earlier in both neurotypical and atypical populations to better characterize them and assess kind of their responses to and how their responses kind of are associated with what we observed in states of pain and distress in the neurotypical and autistic populations from study 1 and study 2. So what we did is we took our equipment, put them in a box, and would send them to the remote destination, their home, or clinical settings, and we would meet with them virtually to guide them through the equipment setup and how to perform the tasks. And then we would obtain their data through cloud-based storage. Cloud-based storage systems. And then the items were sent back to the research lab to be sent to the next participant. So in this study, we had a total of 14 participants, 5 of them being neurotypical and 9 with autism. And 2 of these individuals had a rare SYNGAP, a rare genetic mutation called SYNGAP, which is associated with symptoms of autism as well as intellectual disability. And also the mutation leads to issues with synaptic transmission and excitatory inhibitory imbalances. So looking at these individuals gives us a kind of better understanding of the genetic etiologies of autism and related neurodevelopmental disorders. So we utilized the same kind of biostamp MC10 sensors to capture the cardiac and the motor activity. And then we have them perform the baseline resting task and the pointing task, as well as a 5-minute walking task to assess general gait patterns. All right, so for some of the results, we see the most distinct trends in the motor signal when looking at the pointing task. Specifically, there's elevated gamma variance in the ASD group when looking at movement of the upper arm. And for the sensor on the pectoral area, we see significant changes in the gamma skewness and kurtosis. And overall, this suggests that, in the ASD group at baseline, there's kind of increased variability or noise within the motor signal as they perform the pointing task. And this is consistent with findings in previous studies, as well as what we see during the pointing task in the study 1. Looking at the cardiac activity, we can also assess changes in the HF and the LF powers across time and conditions. So you can see here how for some individuals, there's kind of an imbalance between these two systems that's most distinct during the resting task. And when we look at their cardiac activity, sorry, on the gamma plane, as we observe this pattern of high noise to signal or gamma scale values in the ASD group, as well as the SYNGAP individuals here, whereas the TD group generally demonstrates signals that have lower scale and higher shape values. And there were a few outliers here. So there, A represents a neurotypical individual that had kind of the atypical cardiac response, whereas B here is an autistic individual that demonstrated kind of more of a balance between the LF and HF ranges within the cardiac signal. And so this alludes to kind of the individual differences across the ASD kind of spectrum. And interestingly, when we add the participant with chronic and acute pain, we see that they fall within kind of the same region of the gamma parameter plane as individuals with autism and the SYNGAP mutation. And here's another illustration of the gamma scale and shape and the gamma moments, as well as the heart rate variability parameters, and how we can kind of see distinct separation between the TD and the ASD group. That is clear, more clear here for the Poincaré metrics, as well as the mean, variance, and the skewness. Right. And then lastly, we look at the facial motor activity across both groups. And interestingly, what we generally observed is higher kind of or increased kind of noise across the three kind of regions based on the positional trajectories of each region in the ASD group. And when we look at the transfer entropy ranges, we see that there's significant differences in terms of the influence of, V1 on V2, V1 on V3, and V2 on V3. And the TD group, however, this distinction is not as clear in the ASD group, suggesting that there's a lack of kind of influence on the V1, V2, and V3 regions on each other in the ASD group. When we look at the eye gaze information, we see that based on the positional trajectories, the TD group generally kind of explores more of the 3D space, whereas in the ASD group, there's generally the eye gaze is usually more kind of isolated or restricted to a specific region of the 3D space. And this corresponded with overall lower kind of variability across both eyes in the ASD group. And then lastly, we also look at the facial action units. This graph kind of represents the intensity of each action unit across the entire video frame. So we can see which ones are most intensely kind of expressed, as well as, across participants, which AUs are kind of more present compared to other ones. So for example, here, AU5 is expressed mostly across most participants. And when we do some comparisons between the TD and the ASD group, we find kind of significant differences in the intensity values for the action units. So for example, here, the AU2 was a lot more, or I guess, expressed a lot more in the TD group compared to the ASD group. And this facial movement is associated with the emotion of surprise. So overall, kind of combining all the observations, the results of these 3 studies, looking at externally observable processes such as our movement behaviors, we see that the states of pain kind of induced increased noise and uncertainty in the motor code, which is often associated with kind of increased uncertainty and noise within the motor signal, and with consistencies with task performance, as well as what we observe in the ASD group at baseline. And then looking at the cardiac activity, we observe kind of that both the pressure and the thermally induced pain lead to kind of sympathetic or overdrive or inhibition within parasympathetic activity. And in some cases, at baseline, we observe this in the autistic individuals. And the strong correlation between the SD2 and the gamma NSR metric has been useful in kind of differentiating between the 2 groups in our samples. And then lastly, when looking at the facial expressivity patterns, we see kind of a lack of distinction across the trigeminal kind of V1, V2, and V3 regions in the ASD group. Generally kind of more restricted eye gaze movements and decreases in some of the facial action unit intensity values. So looking at these kind of biorhythmic signals simultaneously gives us a better idea of kind of the overall internal physiology of the individual and how that can impact their behaviors. So lastly, we propose kind of an approach to assess these approaches, our biometric analytics in larger scale populations utilizing commercially available devices such as smartwatches that can track cardiac activity as well as movement activity. Smartphones also are equipped with accelerometers that also can track movement patterns and can be used to capture videos of the face and the body, as well as kind of self-report assessments. And these can be combined to capture all of these biorhythmic signals together, and applying our biometric analyses, we can create kind of an approach to detect, based on the individual's baseline biorhythmic activity, whether they may be experiencing internal states of pain or distress, and possibly kind of alert parents or physicians or teachers when something may be wrong. And this can ultimately lead to kind of early intervention and treatment. Great, so that is all. Here are some of my references. And lastly, I would like to thank my advisor and all members of the Sensory Motor Integration Lab, friends and family, our collaborators, as well as our funding sources that made all of this work possible. That is all. Thank you guys for listening.